Okay, this is prep number 2. I had just finished talking about the different kinds of selection that we use when we're working with bacteria. You have basically nutritional markers. These are, we call them markers because they're genes we use to sort of figure out where things are on a chromosome. So they're like little tags or labels or something like that. And these are ones where if the organism is mutant, it can't make its own, and it requires the addition of the nutrient as a supplement. These ones are carbon source or sugar utilization, and so you provide them a particular sugar, and if they're mutant, they can't actually digest it. It's like being, I don't know, lactose intolerant, I guess, in sort of a way. And if you're wild type, then you can utilize it. So when we plate on a plate that has agar with that sugar, then only the ones that are wild type can grow. And then antibiotic resistance or sensitivity, where the resistant ones can grow, whether there's, the antibiotic is present or not, but the sensitive ones can only grow on a plate that doesn't have any of the antibiotic on it. And this is just an example. So we've got a set of liquid cultures, and you've got a bunch of sensitive, strep sensitive bacteria, but you are looking for, remember I said we can work on a very large scale, so there will be a small proportion of mutations in a particular gene that will allow the cells to grow in the absence, or in the presence, sorry, of streptomycin. And so we're looking for those rare mutations. And so there's lots and lots of cells in here, whoops, but we plate them on generally a lot of plates because, this, as I said, it's very rare, maybe 1 in a billion kind of rare. And so we plate them on a large number of plates, and then we are able to select, because only the ones that actually have that rare mutation are going to grow. All the other, you can see these other little dots, all the other little dots on the plate are the sensitive cells, and those ones won't grow. So this is a wonderful way of working on a very large scale, and only the kinds of genotypes that you're looking for will grow. And so this is another example of looking for spontaneous mutations. There's lots of other reasons we do this, and you'll see when we do the gene mapping work. But in here we've got a suspension of auxotrophic mutations. They're not able to make their own adenine, and there are a few spontaneous mutations. They're called revertants, and these ones, the mutation that prevents these ones from making their own adenine, a nucleotide in this case, that is reversed. So the nucleotide that's wrong in this, the genes of these guys, is spontaneously mutated, and it goes back to the wild type. They're called revertants because they go from being mutant to going back to being wild type. And again, there's tons and tons of cells in here. We might have to plate, 50 plates or something with lots and lots of bacteria on, but there will be these occasional revertant mutations, prototrophs, and if you grow them on media that doesn't have any adenine added to it, then you're going to just only get those ones growing. So again, it's just a way of explaining partly how you do the plating and how that works, and how you select for just certain genotypes, but also to make the point that the kinds of things that we're selecting for are sometimes incredibly rare events, and it's only because of the way we can do this selective screening that we can work with this. Imagine if we had to test like billions of colonies to find out which ones were a certain genotype. That's not practical. You've got to have some way of getting rid of all the ones you don't want and only focusing on the genotypes that you're interested in. So, and it's very efficient. Okay, and we use it a lot, actually. Those of you who might be interested in going on kind of in the genomic kind of field, in genetic engineering, we use bacteria all the time to grow up the plasmids we're interested in, and to make constructs that are going to be used for lots of different purposes in bacteria. So understanding replica plating and things like that will be useful to you for later courses that you may take. So the first little section, I want to talk about bacterial conjugation. It was the first method of gene transfer that was understood at all and was discovered. And Lederberg and Tatum, or Lederberg, I guess it is, basically were working with bacteria, and they had two different strains. And remember that for a lot of these organisms, we knew a lot more about their metabolism, the way they utilized nutrients and things like that, long before we understood anything about their genetics. And so there were two different strains, A and B, and they were both auxotrophic strains. That's what this mutant, not mutant, this minus sign means. And these guys, the type A ones, were mutant for methionine and biotin, so they couldn't make their own of those, but they could make their own threonine, leucine, and thymine. The B types were the exact opposite. They were prototrophic for methionine and biotin, so they could make their own biotin and methionine, but they could not make their own threonine, leucine, and thymine. And so they were doing lots of different kinds of experiments, and they mixed these two together, and they grew them in culture, and then they plated them on minimal media. There shouldn't be anything growing on these, but there were some progeny that grew. And the only way they could possibly have grown is if they had wild types for all 5 of these genes. So therefore, some of this DNA and some of this DNA had somehow combined in some way. There must have been some kind of exchange of materials between those two kinds of bacteria. And so, you know, one of the first questions they had come up with, because I mentioned when I was talking about the plating that there are sometimes what we call revertant mutations. So a mutation occurs that returns a mutant gene back to wild type. It's incredibly rare, but when you're working with the large scale of bacteria, then we can detect them. You know, these rare events that you could never find in a eukaryote. And so what if this was nothing to do with exchange, genetic exchange from these two types of bacteria, but instead very few of them had a mutation back to wild type in biotin, and some others had a mutation back to methionine wild type, and then in a very, very few, you got spontaneous mutations in both of those genes. And so in order to do that, they worked at very, very large scale. And they plated, what is this, 100 million cells. And the kind of frequency of prototrophs that we found in this experiment was approximately 1 in 100,000. Is that right? 1 in 100, 1 in 10 to the 10 million, 1 in 10 million. And so they did experiments on a scale where they plated 10 times as many cells as you would have needed to find those prototrophs in just the A strain. Then if you see any colonies growing, you know that those are revertants because they didn't mix with any of the other genotypes. Same with the B strain, grow up tons and tons of them and just allow them to grow in the rich media. And then when you plate them on minimal media, look for those prototrophic revertants, and there were none in either of these two plates. And so they concluded that it required the mixture of the two different types of cells in order to get this set of prototrophic mutations that seem to combine the genes of both of the strains. And so genetic exchange must be the way this is occurring. And then their next part, this is a weird looking slide because I'm using this picture for a clicker question that I'm going to ask you about. Basically, they decided to repeat it with large amounts of strain A and large amounts of strain B in a big flask with liquid media. And they basically grew them in the rich media, and they had this barrier, and it was a special filter, and the filter had little pores in it, but the pores were too small for bacteria to actually get through. And so the question they were trying to ask here is, do the cells actually have to come into some kind of physical contact in order for this genetic exchange to take place? Or is somehow there's the genetic material, the DNA, is being let out into the medium, into the fluid by some of the cells, and then somehow it can be taken up by the other cells? This is what they were testing. And if this was true, then you might expect some of the DNA might make it through the little pores, or whatever the substance is, through the pores of this filter and make its way to the other side. But what happened was they did this, and then they tested them on, after this experiment, they then put them all on plates that didn't have, that were just minimal media, and looked for colonies, and there were no colonies. Right? So their conclusion here was, it's whatever is mixing between the two strains, it's not just loose, genetic material. It seems to require that the cells physically contact each other. Okay, and it might seem obvious, when you think about it now, you might think, well, how else could they exchange genetic information? But, remember at the time, we didn't know a lot about these processes. And so, and very little was understood. It wasn't even known that bacteria could conjugate or even pick up DNA from the environment under other circumstances and other kinds of bacteria. So this was a reasonable question to ask, and the answer they got was no. For what we're talking about, for this process, physical contact is required between the cells. Okay, so subsequent studies, now when they worked on it some more, they found some interesting things. So for example, there were two types of cells, donor cells and recipient cells. And it turned out that after first of all, the genetic exchange is unidirectional. So it's not like the two cells are kind of swapping information. One cell donates, transfers some of its DNA to the recipient cell, and the other one receives it but doesn't provide anything in return. So it's a one-way kind of transaction. And they called the ones that were donors F+ cells. F stands for fertility. They were thinking of it in terms of being fertile, sharing your DNA, etc. But what's really interesting is that at the end of the experiment, then all the cells were donors. So, and that's kind of an interesting thing, and it's related to kind of how the whole process worked. And then they did notice that donor cells could occasionally lose the ability to act as donors, could become recipients, but that was a really very unusual and very rare event. And I think we have some pictures coming up. Oh, okay. In a minute, we have some pictures coming up. So it turned out that those F+ cells contained a plasmid called the F factor. And that factor is what it has on it, all the sequences that are needed for the production of the pilus, which is a little tube between the two kinds of bacteria, and the transfer of the DNA into the recipient cell. And the plasmid, when it goes through transfer, is transferred completely and is kept by the donor. So the donor provides a copy to the recipient but keeps a copy for itself. And it's a replicative process. Now there was, they started studying a little bit further, and they found that transfer of genes on the bacterial chromosome, which is much bigger than the plasmid. Remember, like you've got your bacterial chromosome, and then the plasmids are corresponding, there's a huge size difference. So this is transferred easily and transferred completely. The bacterial chromosome is not transferred very often, and it's not transferred completely at all. It is a very different mechanism, and we will talk about that shortly. Okay, so we're going to look at a few images. So the F factor It has its own little origin of replication, so it can replicate, and some plasmids have multiple copies in bacteria, but the one we're talking about in this case just keeps one copy. So whenever the bacterium is about to divide, it replicates its bacterial chromosome and it replicates its plasmid, and then the cells that result, the two daughter cells, each one gets one copy of the bacterial chromosome and one copy of the plasmid. And that's what it means when it says replicating autonomously and existing separately, right? And this is how the experiment we're talking about worked. So it has some genes on it. The genes recognize a recipient cell. They form the little tube that the DNA is transferred through, and then they have also the mechanism for that DNA transfer to take place. The recipient cells do not have the F factor, but they do receive it in the process. Okay, and so the way this actually works is it's hard to explain. It's called rolling circle replication. So what happens is the DNA opens and one of the two strands is fed in to the recipient cell. And as it's coming through, the recipient cell goes single-stranded. No. And it starts adding nucleotides to make a double-stranded DNA. In the meantime, this circle keeps kind of twirling. Think of a roll of paper towels, for example, and one sheet of paper towels gets fed until the whole entire single strand has been fed into here. But as this kind of rolls around, then this bacteria fills in the nucleotides for the other strand. So both of the cells, the recipient, or the donor, it keeps one of the strands and then immediately makes double-stranded DNA for the rest of it, and then it feeds one of the strands into the recipient, which then makes it double-stranded immediately, and then it circularizes. So it's just a very weird process, but it's super efficient, because now both of the cells, the donor and the recipient, both have the F factor, so they've both become donor cells. Okay. Now, chromosomal genes can be transferred too, and it's incredibly rare. But if there's a derivative form called the HFR strain, I was just looking for HFR in my notes here, and it's called high frequency recombination. And it's kind of interesting because this was a kind of an accident that caused this strain to form, and it caused some chromosomal genes, as I said, to be transferred a lot more efficiently, a lot more effectively than we would normally see. And in this, when they monitored it, they also found that the F factor was not transferred, and the ability to act as a donor was also not transferred. So it was the actual chromosomal genes from the bacterium that were being transferred into the recipient cell, not the F factor. And so this was really weird. It was called an HFR strain, high frequency recombination. Recombination because once the DNA gets into the bacterium, there has to be crossing over that integrates the new genes into the bacterial chromosome. And it turned out that every now and then, through an accident, the F factor integrates itself into the bacterial chromosome. And now the whole bacterial chromosome acts as if it was this gigantic plasmid. And because it's so huge, the plasmid, when it is a donor cell, it encounters an F- cell, it will try to transfer itself into the recipient cell, but the F factor is right at the very end of the process, and bacterial genes get transferred first. And that's where we're going to start in the next prep.